Alrighty. Hello, everyone, and welcome to Cafe Scientifique from all of us here at SFU's Faculty of Science. My name is Stevie Benish, and I am the Manager of Outreach and Engagement here in the faculty. And if you're one of our cafe regulars, welcome back. It's great to see you. And if you're new, thank you for joining us for the first time. This is our first cafe of 2026, and we are very excited to be kicking it off with such an amazing speaker. So, for those of you who don't know, Cafe Scientifique is the Faculty of Sciences public seminar series where our presenters share their current scientific research and its impact for our local communities and society at large. We typically host about 6 of these discussions a year, and they're all virtual, and they're also all recorded. So if you happen to miss one, you can always go back and view it later, as well as share this one or others with friends and family. And we'd also love to hear from you and stay in touch. So in the post-event email, there'll be a link if you're not a part of our mail list, and you're always welcome to reach out via email. So before we get started, I'd just like to start us off on a good note by acknowledging that I'm very privileged to be joining you today from the unceded and ancestral lands of the Musqueam, Squamish, Tsleil-Waututh, and Coquitlam peoples. We thank them for having cared for and being stewards of these lands and waters since long before our time. And we look forward to keep working with them in a partnership as we continue to build and grow our communities together. And if you know whose territory you're currently on, I would like to invite you right now to take a moment just to reflect on this. And if you don't know whose land you're on, I'd like to share a great resource that you can find out more information. And it's the website native-land.ca, and you can familiarize yourself with the Indigenous people whose land you live and work on. So before we get to our speaker, I just have a couple little house rules to go over. So first note, as I mentioned, this is being recorded and will be shared. There's also a live transcript that is available for your convenience. So following the talk tonight, there will be a Q&A section, but if you do have questions throughout the talk, please feel free to virtually raise your hand and we'll try to get to those. And you can also drop any questions you have in the chat box, and I will try and direct those to our speaker at the appropriate time. So yeah, as I said, please use the raise hand function, which you can find under React at the bottom of your Zoom window there. And when you raise your hand and we call on you, you won't be able to unmute yourself, but we'll be able to unmute you, and you can speak directly to Lisa at that time. I would like to ask, if possible, if you can rename yourself if you don't have a display name right now, so we know who you are when we're having that discussion. And we would also love to see your faces, so I would like to invite you all to turn on your cameras throughout the whole talk, but especially during the Q&A section. So, okay, boring stuff out of the way. Let's get to the good stuff. Tonight's discussion. So I'm very pleased to be introducing Dr. Lisa Craig, who is a professor and the chair of the Department of Molecular Biology and Biochemistry. So Dr. Craig's research focuses on understanding the molecular mechanisms underpinning bacterial virulence. So asking questions like how do bacteria assemble, and what are the secret factors that colonize a host and cause disease? So tonight we'll be discussing the hair-like filaments on the surface of many bacteriopathogens and how they behave to allow bacteria to be pulled along surfaces into protective microcolonies, and how internalized substrates such as DNA and bacterial viruses. We're going to hear about these remarkable molecular machines and how we might exploit their activities to treat multidrug-resistant infections. So without further ado, over to you, Lisa.
Thank you, Stevie, and thank you for that lovely land acknowledgement. Okay, all good. You can see it. So, yeah, I would like to also acknowledge the Faculty of Science for putting on these Cafe Scientifique presentations. I think it's really important that researchers who are paid with tax dollars occasionally present their work and kind of justify what that money is going toward. And so I'm presenting, I'm a protein structural biologist, and you're going to see a lot of kind of complex molecular structures, but I'm going to try to talk about them in a pretty high level. And I would like you to put your hand up if something doesn't make sense to you, because I think it's important that everybody follow along. You have it's a diverse audience with many different backgrounds, so I'm not assuming that you are also structural biologists. So I've broken the talk into three parts. I'm going to give you some biology background on antibiotics and resistance, and try to impress upon you the critical nature of this problem of antimicrobial resistance. Then I'm going to tell you in only a few slides, really, that does that describes about 2.5 decades of work on these type 4 pili, these hair-like filaments on the surface of many bacteria. You can see them here coming out of the surface of these Neisseria, gonorrhea, diplococci. And the work that we've done understanding the filament assembly, structure, dynamics, and functions have led us to propose and test and I think show as proof of concept a new strategy for treating a lot of multi-drug resistant bacteria. So let's start with the antibiotics and resistance. Many of you may have heard of a little bit about the discovery of penicillin 19 in the 1920s. Alexander Fleming was a microbiologist, and he was growing bacteria on these petri plates that were covered in a nutrient agar. And so he takes small amount of bacteria, streaks it onto this plate, and then allows it to grow overnight. And overnight, these few thousands of bacteria multiply to many millions, and then you can see these smears on the plate. But he would get contamination from mold just floating around in the air, and the mold would grow on these and grow quite nicely on the plates. And what he observed is that the bacteria did not grow around the mold. The bacteria, there was this zone of inhibition, and he recognized that the mold or fungi, this is the genus was actually penicillium, is what it was called. He realized that these mold were secreting agents that were killing or at least preventing the growth of the bacteria. And so then it took another decade and a half, and the groups of Howard Florey and Ernst Chain to work out ways to produce this, to first identify this agent, and then produce it in large quantities, to do tests on animals to see if it prevented infection or treated infection, and then eventually clinical trials to see if it was effective in humans. And then the US Department of Agriculture took over and was able to mass produce this compound, penicillin, in time for it to be used to treat infections, bacterial infections, by soldiers fighting in World War II. And it was attributed to saving many, many thousands of lives. And so we refer to that age as the beginning of this golden age of antibiotics. And once scientists recognized that a simple little mold like this penicillium could produce this antibacterial agent, maybe others could. And certainly it turns out that they could as well. Bacteria, a number of different bacteria are capable of producing these different antibiotics. And they do so because it prevents their competitors from growing. And so it allows them to grow in this little protective niche where only they can grow, and they kill every other bacteria around them. And so they have more nutrients for themselves. So if we look at this timeline, the early discovery of penicillin, and then from penicillin being introduced and mass produced and mass utilized, you see that a number of other, and you can probably recognize these antibiotics, vancomycin, erythromycin, tetracycline, were identified, a lot of them from bacterial species. And we call these few decades, this golden era of antibiotics. And it was thought that these would just keep coming. But then the discovery void started to slow down, and very few new antibiotics were introduced over the next few decades. Um, the antibiotics that were brought into market were mostly modifications or derivatives of these original classes of antibiotics. And that's because as these new antibiotics are being brought online, resistance is emerging. And so we can see with every antibiotic, here's the penicillin, which was introduced in 1940 or introduced about 1945, even before it was used to treat people with infections on a large scale, penicillin resistant Staphylococcus aureus emerged. And so for each of these antibiotics, very soon after they've been introduced, antibiotic resistant strains emerge. And so the drug companies were put into and still are in this race with introducing a drug. Bacteria emerge that are resistant to this antibiotic, and so they tweak it, they change they chemically modify it, and now it's effective. But then new bacteria emerge that are resistant to that antibiotic. And this is just natural selection. If an infection is being treated with an antibiotic, it's going to kill most of the bacteria that are sensitive to it. But bacteria divide very, very rapidly, and as they divide, they can acquire mutations. And very rarely, one of those mutations might impart some change, say in a target of an antibiotic, that allows that target to still function, but to not bind the antibiotic and be inhibited by it. And so, by treating with antibiotics, we're really selecting for antibiotic resistant strains because those strains are going to outgrow. In addition to mutations, bacteria can pass genes that encode for antibiotic resistance from one bacterium to another. So we're faced with this. If we look at the 20th century, we see this early on. We've got lots of antibiotics being discovered, but the pipeline slowly starts drying up. And convergently, antibiotic resistant bacterial pathogens are emerging, passing this resistance on to other bacteria. And so we've got this real problem where our antibiotic pipeline is almost dried up, and we are facing antibiotic or bacteria that are resistant to multi-drugs, some that are resistant, that are referred to as extremely resistant or extensively resistant. And we even have pan-resistant bacteria that are resistant to all known antibiotics. So this is a real serious problem. And it is estimated that as this problem continues to grow, we keep having antibiotic resistance emerging without a lot of new antibiotics being brought to market, that we're really going to increase. 2021, there were about 4.7 million deaths associated with antibiotic resistant bacterial infections, and that's expected to double by the year 2050. And so myself and others recognize this really as an existential threat, a global health threat on par with climate change, with another viral pandemic, and with the disruption in the loss of order-based rules, and the disruptions globally that we're seeing with geopolitics. And all of these threats are really interrelated because climate change is altering ecosystems where bacteria grow. They can grow in new ecosystems. Viral vectors can now spread to new environments where that were never hospitable to them before. And of course, changes, particularly that the United States is making with the Trump administration, where vaccines are no longer being recommended, where international aid is no longer available. We're just going to see these problems exacerbated. And I'm going to tell you about a strategy that we have identified that I think has the potential to be quite effective at treating bacterial infections. But in fact, There's nothing that beats preventing bacterial infections and preventing viral infections, and that's what vaccines do. And so the loss of or the problems that the U.S. has created in not recognizing the value of vaccines is really going to just add to this perfect storm that is brewing.
It looks like we have a question from the audience.
Okay, sure. Yeah, I can't see them, so go ahead and interrupt me when that comes up. And if it turns out that there are too many questions, I will get you to leave them to the end. But for now, let's start with just asking questions in the middle of the talk.
Rosemary, please ask your question.
Okay, hi Lisa.
Hi, Rosie.
I appreciate your what you're trying to tell us right now. I just have one little modification, if you don't mind, for you in the audience, because I just read a couple papers on this,
Okay,
and
if I
that
could
is
just say, if I could just say that Rosemary Cornell was my master supervisor, so she's correcting me again.
so 4.7 million deaths due to antibiotic resistance, but climate change, you mentioned this is similar to what we anticipate from climate change.
Oh,
So
no, no, no. I'm not comparing them on a level. I'm just saying
okay,
these are existential threats to humanity.
okay. The number I have here for deaths from due to climate change, not just heat, but all the associated impacts is by 2020, by 2100 is a billion.
Are we
Okay,
competing
enough
here?
said. Continue. We're
Okay.
laughing. It's a displacement behavior.
Thank you, Rosie.
Okay.
So now I want to just give you a bit of biology on what are antibiotics and what is resistance. So antibiotics are relatively small molecules, and as I said, a lot of them are natural products. They're produced by other microbes, and they target particular processes in the bacterial cell. Those might be unique processes. For example, the cell wall. Bacteria make a cell wall called peptidoglycan, and a lot of our antibiotics, including vancomycin and beta-lactam antibiotics like penicillin and methicillin, those inhibit cell wall synthesis. So that's a very unique process for bacteria. So those antibiotics are very specific for bacteria. That's why they can be taken by humans, given to mammals without having any serious side effects. But other antibiotics target processes that are also occur in mammalian cells, like DNA synthesis, RNA synthesis, protein synthesis, but they will target a molecule that is unique in the bacteria. And so that's how antibiotics work. Antibiotic resistance in a bacteria emerges where a change occurs in the DNA that encodes for usually a protein, and then the protein that may be normally bound to that antibiotic and was inhibited no longer binds to that antibiotic, but the protein can still function. So that results in resistance, and that's by mutation as the bacteria replicate. Small mistakes can be made in DNA synthesis. The proteins can change a little bit, and then those resistant strains are selected by antibiotic treatment. But bacteria can also take on big chunks of DNA that have genes that encode for whole pathways that allow them to resist antibiotics. They can acquire genes that encode for pumps that pump out the antibiotics from the cell, or that provide different pathways that can entirely avoid the antibiotic, or provide genes that encode for proteins that inactivate antibiotics. So there are a lot of different mechanisms by which bacteria can acquire resistance. And as an example here, we'll come back to this cell wall, that peptidoglycan, that all bacteria need this cell wall. And the beta-lactam antibiotics that I'm showing here, penicillin, that first antibiotic that was discovered, methicillin, there are many different beta-lactam antibiotics. Beta-lactam, because they have this beta-lactam ring that's conserved, and then they vary in other parts of the antibiotic. And so bacteria can acquire a gene that encodes a beta-lactamase to degrade that, to break this bond and inactivate that penicillin. Peptidoglycan is formed by proteins called peptidoglycan binding proteins. Obviously, they were named because these enzymes bind peptidoglycan. Bacteria can acquire new PBPs that do not bind that antibiotic that they're being treated with, but can still synthesize the cell wall. So just a couple of common mechanisms for antibiotic resistance. And these for instance, a gene for a new peptidoglycan binding protein or for a beta-lactamase, these genes can be transferred from one bacterium to another. And there are three main mechanisms for transfer of genes from one bacterium. So from one bacteria that has a resistance mechanism shown in red here, a donor cell that is resistant can transfer some DNA containing that resistance marker to a recipient cell via what's called a conjugative pilus. That's called conjugation. Another mechanism is transduction. So bacteriophages are viruses that infect bacteria. And those bacteriophage, when they infect bacteria, they can pick up genes from the bacterium, like antibiotic resistance markers, and then they infect another bacterium and they can transfer that marker. And then the third mechanism, and this is a mechanism that we are exploiting as a strategy, it's called natural competence, where bacteria die, they lyse, they release their DNA, and that DNA can be taken up by neighboring or adjacent cells via this DNA uptake machinery. And some of that DNA may have antibiotic resistance genes encoded on it, and that antibiotic or that resistance can be acquired by that bacterium. So, what are the bacteria that are the biggest problems in terms of antibiotic resistance? Well, every year the World Health Organization puts out a bacterial priority pathogen list, and this is shown for 2024. And they list critical groups, high groups, medium groups, and these bacteria or groups of bacteria, for instance, Enterobacterales, that encompasses a large number of different bacteria that are carbapenem resistant. But carbapenem is a newer antibiotic, and all new antibiotics are held back and used as last resort antibiotics for multi-drug resistant bacteria that are not resistant to the typical antibiotics that we use. And so if you see something that's carbapenem resistant, it is typically multi-drug resistant, resistant to streptomycin, resistant to vancomycin, maybe resistant to the beta-lactam antibiotics. So I'm not going to go through this list, but I do want to talk a little bit about the bacterial physiology and then come back to this list. Because bacteria can be divided into two categories. We call them gram-positive bacteria, gram-negative bacteria. And the gram is just somebody with the last name Gram identified a stain that could distinguish them. And the gram-positive bacteria take up stain differently than the gram-negative bacteria. They look purple. These guys end up looking pink. So you can distinguish them just on a light microscope. All bacteria have this cytoplasm where they have their DNA and all their cellular functions in the cytoplasm, which is encased in an inner membrane. And then they all have peptidoglycan. And in the gram-positive bacteria, that peptidoglycan or cell wall is very thick. Gram-negative bacteria, it's very thin. And there's a second membrane, an outer membrane. And that membrane is an additional resistance. It's a barrier for especially large antibiotics. And so if you look at these high priority pathogens, the majority of them are gram-negative bacteria, which have acquired various mechanisms of resistance, but are also difficult to treat just because of that outer membrane. And our strategy is going to overcome that outer membrane barrier. And so what we are doing, what we've shown is actually pretty effective, at least in the lab. Many bacteria including a lot of gram-negative bacteria and important human pathogens, have type 4 pili. These are hair-like filaments. They're displayed on the surface of the bacteria. They are polymers of what we call the major pilin protein. So thousands of copies of this major pilin that form this filament. And then they also have minor pilins at the tip of the pilus. And the minor pilins pilus assembly starts in the inner membrane. The minor pilins form these nucleation sites that initiate pilus assembly. The major pilins join in. Then the minor pilins end up at the pilus tip. The pili assemble very rapidly, about 1,000 subunits per second. And then they retract. And so they're dynamic filaments. They rapidly assemble, they rapidly retract, and they're adhesive. And so these features are the features that we're going to exploit. So the adhesive nature is often due to the minor pilins, although they're not that well characterized, the minor pilins at the tip. So I like to think about the minor pilins as being the hands of these long arms. They reach out from the bacteria, they grab onto things, and then they pull these things into the bacteria, or they pull themselves along. And so they can bind to receptors on epithelial surfaces. Think about Pseudomonas aeruginosa infecting the lungs of a cystic fibrosis patient. They can bind to, these pili can interact with each other, allow the bacteria to form these large, tight aggregates that protect them from the host immune response and even can protect them against antibiotic treatment. Some type 4 pili, and the functions differ from one species to another. Some type 4 pili can bind to bacteriophage, filamentous phage. I'll talk a little bit about this, or other phage. And then they retract and they pull the phage, the genome enters the cell, and you can get transfer of antibiotic resistance in that way. And finally, some type 4 pili mediate natural transformation. They bind to DNA, they take up exogenous DNA, and they bring it into the cell. The pili bring the DNA right into the periplasm, that region in between the inner and the outer and the inner membrane, and then other proteins will bring the DNA into the bacterium. And so it's this DNA that we are using. We thought that maybe we could attach an antibiotic, one of these antibiotics that doesn't easily penetrate the outer membrane, attach it to DNA, which we know binds to the pilus, and then upon pilus retraction, that DNA antibiotic conjugate gets taken up into the periplasm, and maybe that will kill the bacteria. So again, that's just our little cartoon of natural transformation. And I want to show you a video from our collaborator, Oncordalia at University of Indiana. He's showing a bacterium here, dark green. He's labeled the bacteria and their pili. The pili is assembling. It's grabbing onto DNA. So the DNA is fluorescently labeled red, grabbing onto it and pulling it into the cell. And so this is the mechanism that we're going to exploit as an antibacterial strategy. We call this a Trojan horse mechanism because the type 4 pilus is the soldier who's bringing the Trojan horse into the fort. That Trojan horse is the DNA carrier, then it's going to be carrying antibiotic, and this hapless soldier is going to be leading it in as the type 4 pilus would be guiding it into the cell. So before I go into showing you how we've tested this hypothesis, I want to tell you a little bit about what we've learned over the last couple of decades about type 4 pili that have led us to test out this strategy. So these are some electron microscopy images. Here's Neisseria gonorrhea. They're the type 4 pili. But they're present on a number of human pathogens. Enterotoxigenic E. coli, or ETEC, causes traveler's diarrhea. Acinetobacter baumannii, multidrug-resistant pathogen. So the type 4 pili are present on a number of important human pathogens. I've already showed you Neisseria gonorrhea, which causes the sexually transmitted disease gonorrhea. Um, Enterotoxigenic E. coli display type 4 pili. This causes traveler's diarrhea. Acinetobacter baumannii is an opportunistic pathogen that causes wound infections, urinary tract infections, a lot of different infections. Pseudomonas aeruginosa infects wounds, burns, and it's the major killer of people with cystic fibrosis. Vibrio cholera causes the diarrheal disease cholera. And all of these organisms, if you delete genes encoding their type 4 pilus machinery, they are no longer pathogenic. So type 4 pilus functions are essential for pathogenesis. And I already talked about some of those functions. Natural transformation, twitching motility. So that's where the bacteria adhere to a surface and then can retract and pull themselves along. That's important for dissemination. This microcolony formation, that's when pilus-pilus interactions hold bacteria into tight aggregates that protect them. They can adhere to host surfaces, take up phage, and they can also secrete, some type 4 pili secrete substrates from the bacteria into the extracellular space. We've worked a little bit on that as well. Okay, so type 4 pili, these are these filaments. They grow from the inner membrane of gram-negative bacteria, and they're present on some gram-positive bacteria, so they also grow from that same membrane. We call it the plasma membrane. They grow across this periplasm, across the peptidoglycan, through a secretin channel in the outer membrane, and then they're displayed on the surface. And so I'm going to be showing a cartoon like this for lots of the slides. Just take you through it. So now here's the inner membrane, here's the outer membrane, and that secretin channel. The type 4 pili grow from these major pilin proteins. These major pilins are anchored in the inner membrane. This is a model for assembly. They're thought to diffuse to the base of the pilus. Then you've got an assembly motor that talks to some proteins in the inner membrane and probably causes an incremental extrusion of the pilus, which now then opens up a gap on the other side for another pilin subunit to incorporate. So they grow from the base up, and then they retract just in the reverse of that mechanism. And so I would call myself a structural biologist and a microbiologist, and I've been interested in protein structure since I started my master's in Rosemary Cornell's lab here at Simon Fraser University and continued with Jamie Scott, also at SFU. And then I went to do a postdoc in John Tainer's lab at the Scripps Research Institute in California, where I started to work on these type 4 pili using structural biology, or using X-ray crystallography and cryo-electron microscopy to understand the structure, the atomic structure with X-ray crystallography, and then the filament structure using cryo-EM. And just to say a few things about protein structure or proteins in general, because not everybody listening is a protein biochemist. Proteins are long chains of amino acids. We have 20 different amino acids, and each amino acid has a different character, a different size, a different chemistry. And the sequence of those amino acids dictate how that protein will fold up into a three-dimensional structure. And then the amino acids on the surface will dictate how it interacts with other substrates. And so myself and my group have solved a number of structures of type 4 pilin, these major pilins, and they all have this canonical structure. And here I'm just coloring the carbon atoms in red and the nitrogens in blue, sorry, the oxygens in red, the nitrogens in blue, the carbons in gray. And this little alpha helical stalk, the character of the amino acids, you can see there are not a lot of oxygens and nitrogens. These side chains that are poking out, they give it a hydrophobic character, which means that this region does not want to interact with water, the aqueous environment, but it's very happy in this inner membrane, which is a greasy, oily interface, and it's very happy in the core of the filament interacting with other hydrophobic alpha helices. And so, let's see, I think the next slide is a lot of our cryo-EM structures where we've solved the structure of the pill and subunits that's assembled in this helical filament. And they all seem to assemble in essentially the same way. If we just look end on, you can see that these, what we call the globular C-terminal domains, are out there on the surface, and that's what's going to form the surface of the filament. And then in the core of the filament are these hydrophobic alpha helices. Um, the type 4 pili can be classified into, they're really a continuum, but I like to think about them as simple or complex type 4 pili. So I want to introduce a few more characters in this pilus assembly. In the complex type 4 pili, not only do they have these major pilins that form the filament, I'd mentioned minor pilins. And they're called minor pilins not because they're smaller, but because they're much lower in abundance. And the minor pilins are thought in the complex type 4 pili to assemble into this cluster that then initiates nucleation of the major pilins, which means that those minor pilins end up on the tip of the pilus and they can mediate functions like adhesion and interactions with other substrates. And there are multiple minor pilins in the complex type 4 pili that are seen in Neisseria gonorrhea, Neisseria meningitidis, Pseudomonas aeruginosa, etc. I mentioned the assembly ATPase that's critical for assembling the pilus, but these complex type 4 pili also have a retraction ATPase. It's a second motor that's needed for pilus disassembly. So assembly ATPase causes assembly, and then maybe they switch on and off. We really don't know the mechanism, but the retraction ATPase is required for pilus retraction, and so to get a dynamic pilus. If you knock out the genes and the gene encoding this retraction ATPase, these bacteria just make really long pili, and they're not functional because they can't retract. Simple type 4 pili, they don't have a retraction ATPase, and so it was thought that they were not retractile. They would just grow. They have a single minor pilin. They don't have this nice cluster. And when we started exploring this, we didn't know what the function of this minor pilin was. Does it also initiate pilus assembly? Well, we didn't have a structure for it, but based on the amino acid sequence, as you can see in my cartoon, it's a lot bigger than the major pilins. And we predicted that it would assemble just like a major pilin, but it would have this C-terminal end, this big end, and we thought that might contribute to pilus retraction. I'll take you through that model in a minute. So we wanted to know, do these simple type 4 pili, do they actually retract even though they don't have a retraction ATPase? If so, how do they retract? We suspect the minor pilin initiates retraction. And is retraction required for type 4 pilus functions in Vibrio cholera and ETEC and these other simple type 4 pilus systems? So what are the functions of type 4 pili in these simple systems in Vibrio cholera and also in enterotoxigenic E. coli or ETEC? One of them for both of these organisms, the pili stick together. You can see these nice bundles of pili on the Vibrio cholera cells, and they bring the cells together in these aggregates. And you can grow up Vibrio cholera overnight in liquid conditions, and in the morning, all of the cells have fallen to the bottom in these aggregates, these macroscopic aggregates. And here's a scanning transmission electron microscopy image of one of those aggregates, and it has millions and millions of Vibrio cholera cells packed really tight. And so looking at this, we hypothesized that pilus retraction might be required to pull them into these tight aggregates. The type 4 pili in Vibrio cholera are the receptors for this cholera toxin phage. And as an aside, the cholera toxin phage has genes encoding for cholera toxin. And so many, many years ago, millennia ago, Vibrio cholera that were non-pathogenic floating around in seawater got infected with this CTX5 phage, and the genome incorporated into the Vibrio cholera genome, and they were able to make cholera toxin and become this potent human pathogen. But I digress. My point here is that the CTX phage bind to the pilus, and in order for the phage to get in, the pilus must retract. So I'm building this story that to justify that type 4 pili in Vibrio cholera and ETEC must be retractile, even though they lack a retraction ATPase. Finally, in these organisms, the type 4 pilus is responsible for secreting an exoprotein or a substrate. Both of these, it's a single protein in Vibrio cholera, a different one in ETEC. We've solved the structures of these. We know they're important for colonization, but we don't know what their function is. And again, you would expect that you would need a dynamic system to pump out one exoprotein and then come back and pump out another one. So our hypothesis was that these type 4 pili are retractile. And we suspect that this minor pilin induced retraction. And so to first show that they're retractile, we worked with Nicholas B.A. He was at Brooklyn College at the time, and he developed this micropillar system, these microscopic little templates or grids that had these pillars that are elastic and flexible. And you could allow bacteria to settle on top of them, and the pili would grow and adhere to the adjacent micropillars. And then when they retracted, they distorted these micropillars. And so if we're going to look at a video, we're looking from the top down through the slice of the micropillars. And so you can see a few micropillars here. We're looking from the top down. When I play this, you'll see bacteria darting around, but they move very quickly. But some bacteria have settled, and you can't see those bacteria. What you can see is the micropillars moving. And that's because the pili have settled, they're retracting, and they're pulling these micropillars together. And I can just slow this down, and you can see those two micropillars moving back and forth. And if we delete genes encoding for the type 4 pilus, then we lose this activity altogether. And so that told us, yes, these Vibrio cholera are indeed, the pili are indeed retractile, even though they lack a retraction ATPase. And so our model, I'm going to stop that, was that this minor pilin acted like a major pilin. Remember, it's minor, so it's very low abundance. So you're going to get assembly of the pilus, and then occasionally a minor pilin is going to dock in, pretending it's a major pilin. But this large C-terminal domain is going to sterically block passage of the pilus through the outer membrane secretin channel. I haven't showed that here. And so that was our model. And so then we reasoned that we could just delete the gene encoding the minor pilin, and then we should get nice long pili, but no retraction. In fact, what happened is we got no pili at all. And so what that told us is that like the complex type 4 pilus systems, the minor pilin is initiating pilus assembly. And so we actually solved the structure of this minor pilin. We predicted that it would form this monomer at the tip of the pilus. Then we did some electron microscopy to try to image the minor pilin. And we took antibodies, an antibody that's specific, that binds very specifically to this minor pilin, and you can purchase another antibody that will bind to that antibody that has a big gold label, and so the gold electrons do not pass through that gold. So that's what we were looking at, is we have an antibody binding to our minor pilin. It's got this big gold label, and we're able to see these golds, and the golds tell us where our minor pilin is. So these are our pili, and hopefully you're not looking at this on a phone, or you won't be able to see it. But in some cases, you can see these clusters of minor pilins at the tips of these pili. And we can see as much as 3 minor pilins, which told us And this information was complementary to X-ray structures that we determined for the minor pillins for both E-tech and for Vibrio cholera. In the crystal structure, they formed a trimer. And so we've modeled the minor pillin as a trimer at the pilus tip. So far, here's our model, is that these minor pillins associate via their C-terminal domains. Remember, these are the pillin domains, but those C-terminal domains, that's what holds the proteins together. And what I'm showing you is exactly what we saw in the crystal structure. C-terminal domains bring them together. That forms a minor pillin priming complex that then can nucleate or attract the first minor pillin. Then you get extrusion, and then another minor pillin comes in, and the pilus grows up through that secretin channel. But we still felt like this minor pillin had a second role in addition to its role in initiating pilus assembly, that it could initiate pilus retraction. Something had to initiate pilus retraction, and we don't know they don't have a retraction ATPase. So we predicted that it would be this minor pillin that was doing that again, by the steric hindrance model. But if we knock it out, we just lose pileation, so that doesn't tell us about its role in retraction. We took advantage of a conserved residue, glutamic acid. That's an amino acid, and it has a negative charge, and it's present on all type 4 pillins, both the major pillins and the minor pillins. And it's critical for the major pillin docking into the growing pilus, because there's a positive charge at the end of every pillin. And so this is an electrostatic attraction that occurs. And that's part of driving this pilus assembly. If we change that residue in the major pillin to something that is not negatively charged, we lose pilus assembly. So we know it's important for pilus assembly. We hypothesized that it would be important for this function, for the minor pillin docking in to initiate retraction. But that it would not be important for priming pilus assembly. And that's because it's those C-terminal domains that help to form that priming complex. And we hypothesized that this would still occur without GLU5. So we made some substitutions in that GLU5. And sure enough, what we found, oh, there's my GLU5, is that with the GLU5 mutants, the pili are assembled, but they're no longer retractile. In that assay that Nicola Ba does, he no longer saw micropillars moving, so they're no longer retractile. And also, all of pilus functions are disrupted. That is, pilus aggregation, cholera toxin phage uptake, and secretion are all disrupted. So that tells us that pilus retraction is required for pilus functions. So here's our model for how the minor pillin induces pilus retraction. Is we've got it docking into the pilus, and then as the pilus grows, this C-terminal domain blocks passage of the pilus through the secretin channel. That causes pilus assembly to stall. This last subunit that got added is no longer stable when the pilus isn't growing, and so that subunit diffuses away, and you get a collapse. And then the next subunit diffuses away, and you get this spontaneous collapse, and then the pilus has to reassemble. So now we had a good idea about the pilus dynamics in Vibrio and in E-tech, and we knew that the minor pillin forms a trimer at the pilus tip, and that it also initiates pilus assembly. We went on to show that that minor pillin is actually the receptor for the cholera toxin phage. We purified the tip proteins, the phage tip proteins, and showed that they bind directly to the C-terminal domain, this trimerization domain of the minor pillin. And then we showed that the minor pillin also mediates secretion. So we solved structures of these substrates, TCP-F in Vibrio cholera, and it had this flexible N-terminus, which we showed associates with the minor pillins and then is delivered across that outer membrane, released, pilus retracts, grabs another one, and secretes it. So all of this, many years of work understanding pilus structure, pilus assembly, pilus functions, got me thinking that we could exploit this dynamic and adhesive nature of type 4 pili. And we thought that that DNA might be the best substrate to use as a carrier for antibiotics because we know that we know it's something that we can easily make, and that it binds to a lot of different type 4 pili or type 4 pili and a number of different pathogens. So to see if this would work, we used a model system, which is we use Neisseria gonorrhea, gram negative. They are resistant to large antibiotics, including vancomycin, because they have that outer membrane. They have type 4 pili, and those type 4 pili make them naturally competent. So those type 4 pili bind DNA. The antibiotic of choice, and this is not to scale, obviously, is this large antibiotic vancomycin. It's known as a last resort antibiotic because it's kind of a nasty antibiotic that has some toxicity. The function, like beta-lactam antibiotics, like penicillin, is it inhibits cell wall synthesis. So we only needed to get into just across that outer membrane to inhibit cell wall peptidoglycan synthesis right in the periplasm there. And we figured the type 4 pilus could do that for us. Vancomycin is very effective against gram-positive bacteria, but they don't have that outer membrane. And then we needed a DNA carrier. And this is where we brought in Dipankar Sen, who is a colleague in the molecular biology and biochemistry department, and he is a DNA aficionado. And he gave us an oligonucleotide that was a 79-mer that had some catalytic activity, and he's identified that showed that DNA molecules can actually perform catalysis like proteins can. So he gave us a 79-mer, a double-stranded DNA, and he said, try this. And then he helped us. Shivani Kundra, who was a postdoc in my lab, worked to used various different attachment mechanisms, finally landed on a process called click chemistry to attach vancomycin to DNA. And here is a curve showing the survival of Neisseria gonorrhea. So she treated Neisseria gonorrhea in solution. If she uses just free vancomycin, that's our black line here. She's got to add 84, a high concentration, 84 micromolar free vancomycin in order to kill these Neisseria gonorrhea. So we're going from about 100 million cells, 10 to the 8th, down to our limit of detection of about 100 million cells. So that takes a lot of free vancomycin. If she uses the DNA vancomycin conjugate, she can go way lower to 6 micromolar, 15-fold reduction. And this is if both the double-stranded and the single-stranded are effective in killing Neisseria gonorrhea. So now she's bringing it into a concentration that actually is serum achievable, that is possible that we could use this conjugate to actually treat Neisseria gonorrhea infections. So then we wanted to make sure, is this because the type 4 pili are bringing it in? Well, if we look at our wild type cells that make retractile type 4 pili, we add the DNA vancomycin conjugate, and at 0 time, the cells, we have 100 million cells, and then after 4 hours, we cannot detect any live cells anymore. So that's what I just showed you in the previous slide. But now if we look at Neisseria gonorrhea that don't make pili, at 4 hours, we see no killing by this DNA vanc conjugate. And if we look at a mutant that makes pili but are non-retractile, we also see no killing. And so that tells us that this DNA uptake, the killing by the DNA vanc conjugate, is pilus mediated, and it requires retractile pili. We looked at, okay, can we make this smaller? The 79-mer is a pretty big oligonucleotide. Well, we know that we can at least cut it in half because single-stranded DNA is just as good at killing as double-stranded is. We can make it longer, and it's great at killing. No better, but just as good. We can't make it shorter. made a 70-mer, so it just took 9 nucleotides off. Very poor killing. Make a 60-mer, we get zero killing. So fortuitously, we had started with sort of the minimal length of nucleotide to be effective as a vancomycin carrier. A couple other things. The sequence of the DNA does not matter except for this particular DNA uptake sequence that had been characterized by a number of other groups that is needed to bind to the type 4 pilus. She showed that not only is this conjugate effective in killing lab strains of Neisseria, which many years ago were isolated by patients but have been passed in the lab for a long time, but that it's also just as effective at killing patient isolates. And so Linda Huang at the BC CDC sent us a bunch of Neisseria gonorrhea patient isolates, and they all kill. So here's our DNA vanc conjugate at 6 micromolar. Here's vancomycin at 6 micromolar. We see no killing. Vancomycin at 86 micromolar. We have to go up to 86 micromolar to see killing. And we see that with every single one of these patient isolates. We also see that the DNA vanc can kill other bacteria that are naturally transformable. Acinetobacter bailii is naturally, this is a bacterium that's not a pathogen, but it's heavily used to study natural transformation. It's highly transformable. It takes 14 micromolar DNA vancomycin to kill Acinetobacter baumannii. 14 micromolar does, Acinetobacter bailii. 14 micromolar does nothing when it's just free vancomycin. You have to go up to 170 micromolar free vancomycin to kill. Acinetobacter baumannii, as I mentioned, this is a serious pathogen. This is one of the critical high priority pathogens, and it is multi-drug resistant. We can kill that with 20 micromolar DNA vanc. We have to go up to 650 micromolar free vancomycin to see the same level of killing. So that's really promising that we can use this in other organisms. And then finally, we moved to sort of a next step to animal testing is we used a wax moth larva, Galleria melanella. And this has been used as a gonorrhea infection model. These moths succumb to Neisseria gonorrhea infection. And so we first tested our DNA vanc conjugates at multiple concentrations, as well as free vancomycin and just a buffer and single-stranded DNA alone. And we find that while free vancomycin is somewhat toxic to the cells, so here we're plotting the survival of these worms after injecting one of these reagents over 3 days. And we see that free vancomycin in red here is toxic to the cells. After 3 days, 20% of the worms have died, but we see no toxicity with various concentrations of our conjugate. So then we moved into an infection model where, and this is all work again done by Shivani Kundra. She injected the worms with Neisseria gonorrhea, and then 30 minutes later, she treated them with our various conjugates. And I won't point out all of these, but I will show you that, let's see, buffer treatment, or let's say no treatment because I can see the black. Here's no treatment. After 3 days, most of those cells, 90% of those larvae die with no treatment. So they're being killed by the Neisseria gonorrhea. If she injects them with free vanc at the highest concentration, here's the red line, very little efficacy in protecting them against the Neisseria gonorrhea infection. However, the DNA vanc conjugates, when she goes up to 5 or 7 micrograms, she's getting 85 to 90% survival. And so this is an invertebrate model, but it's proof of concept that this DNA conjugate is effective. So I want to just summarize our findings from the last half of this talk is that we were able to attach vancomycin to a DNA carrier, which substantially increased its bactericidal activity for Neisseria gonorrhea lab strains, but then also for a number of patient isolates, as well as other bacteria that are naturally transformable and that is effective in protecting the wax moth larva Neisseria gonorrhea infection model. It's effective at protecting it against killing by Neisseria gonorrhea. And so we think that the DNA associates with the type 4 pilus. It maybe wraps around the tip and that's how and sort of gets compacted and that's how it's able to be brought into the periplasm here. Then it's released. There are other proteins involved in its release, but it has this vancomycin that then can inhibit this penicillin binding protein, the enzyme that synthesizes peptidoglycan. And so it inhibits that and it disrupts cell wall biosynthesis and kills the bacteria. Future directions, where are we going next? Obviously, there is not a global health concern about wax moth larvae being infected with Vibrio cholera or with Neisseria gonorrhea. We want to move this into animal models, starting actually with just cervical epithelial cells and seeing if it protects them, going into mouse models. We want to increase the payload. We've just got one antibiotic attached to the 79-mer single-stranded or double-stranded. There are ways that we can modify, that we can incorporate it throughout this, and we'll have to test to make sure that that doesn't disrupt DNA binding or binding to the pilus and uptake. We need to modify the DNA so that it has optimal pharmacokinetics so that it can survive when injected into a human. Looking at other naturally transformable bacteria, is it effective against these? Is it effective when bacteria are in biofilms that tend to make them resistant to antibiotics? And can we get other antibiotics, attach other antibiotics to DNA and get them to work? And so just lastly, I want to very quickly acknowledge some of the many people who've worked on the results that I've talked about here, in particular the DNA conjugate, Shivani Kundra, who came as a clinical microbiologist and had to learn chemistry and has done a really pretty remarkable job. She has essentially done all of this work. Dipankar Sen, in 2018, I was running out of grant funding and I told him about this idea and he got his lab behind it and made this happen with his collaboration as well as that of Linda Huang at BCCDC. We're able to get funding and it's just taken off from there. And Kunlu has been tremendously helpful in just purifying our DNA conjugates because there's some toxicity if we don't purify them sufficiently. Lots of folks from my lab over the many years who've contributed to every pillin structure that I've ever solved was solved by Supermania Colopan and wonderful collaborators over the years, in particular Ed Egelman, again, behind every cryo-EM structure that has ever come out of our lab. And as well, acknowledging the funding agencies, NSERC and New Frontiers, as well as CIHR. And with that, I will end the presentation. Thank you.
Wonderful. Thank you so much, Lisa. I was thinking about what you said at the top of the presentation about how important it is for researchers to share their work with, the communities that they're a part of, and I think you did an excellent job of that tonight.
Thank you. I hope it was understandable to at least some people. I don't know how long I went. I feel like I went kind of long. It's 6:27.
That's okay. I was wrapped. It was fascinating. I I think I
Well,
say this every time, but I love doing these talks. It's such a great part of my job because I get to learn such incredible things from such incredible people. I do have just a couple quick questions before we
sure.
wrap up, if you don't mind. Something I always like to ask is in kind of the process of this research, which was the thing that was the most surprising to you? Like, what really caught you off guard in your research?
Hmm. Um, I What really catches me off guard is when I have a hypothesis and I'm able to prove it. Because most of the hypotheses that I come up with, that we come up with, just do never pan out.
Yeah.
And so we've been able to show some really interesting mechanisms, and we did that from a structural point of view, looking at the structure, how might this work? And when they actually did work, like showing that that minor pilin could induce pilus retraction, that was really incredible. Showing that the minor pilin interacts with the phage, that it induces secretion. Those are really amazing. And of course, I haven't talked to you about all of the hypotheses that didn't pan out and all of the experiments that we've done over many, many years that haven't worked.
I mean, that is part of the process of science, but
It
it
is
is
indeed.
very exciting when things do turn out. And I guess my other question before we have to wrap up is just what are you most excited about going forward with this research?
Yeah, I'm super, Shivani and I and Dipankar are super excited about this DNA conjugate because we feel like it has the potential to treat multiple pretty serious human pathogen infections. We see pretty good efficacy with just this one vancomycin attached to the DNA. I think we can do a lot better with that. So we have all kinds of directions that we can go in, and I think that this is something that could actually work as a treatment.
Yeah, yeah, the possibilities are so exciting, and I feel like we're gonna have to have you back for another one of these in a couple years and get an update of kind of where things are at. But I think we've come to our end of our time for this evening, so I just want to say a huge thank you to everyone who joined us this evening, and a huge thank you especially to you, Lisa, for taking the time out of your evening to share this incredible research with all of us.
You are very welcome, and thanks everybody for attending.